أهلا بكم في الموسم الجديد من عالم الوراثة. تابعونا.
Molecular biology and genetics. On behalf of the National Committee for Biochemistry and Molecular Biology and Genetics, Academy of Scientific Research and Technology, it is our great pleasure to welcome all of you to today's lecture entitled Mushroom Bioactive Compound from Food to Nutraceutical, Cosmeceutical, and Biopharmaceutical Industries. I'm honored to introduce our distinguished guest speaker, Professor Hisham Al-Anshasi. Professor Hisham Al-Anshasi, he is the director for Innovation Center in Agri-Technology for Advanced Bioprocessing, Faculty of Chemical and Energy Engineering, University Technology, Malaysia. UTM. Welcome, Professor Hisham, and please you can start.
Okay. بسم الله الرحمن الرحيم والصلاة والسلام على سيدنا محمد وعلى آله وصحبه أجمعين. Actually, it's given me great pleasure and honor for being with you today and invited with the highly prestigious committee for National Committee of the Biochemistry, Molecular Biology and Genetic Engineering under Academy of Scientific Research and Technology, Egypt. Actually, I would like just to share, maybe many of you knows me already since years. And thank you very much, Prof. Abdul Latif Hisham and the Academy for this invitation. And my name is Hisham Al-Anshasi. I'm already 18 years here in Malaysia. Before that, I was the Assistant Director for Research Institutes Affairs in City of Scientific Research, Alexandria. I speak today in English because I have a couple of friends and colleagues from Malaysia, Indonesia, and other countries. When they know that we're having this international webinar, they love to join. I think this is also good as a platform to say Egyptian Academy of Science Technology reaching the world, not only for Egyptian scholars, but also they, and I hope all are going to enjoy for being virtually in Egypt today with us. Okay, my talk today is about the mushroom bioactive compounds from foods to nutraceuticals, cosmeceutical, and biopharmaceutical industry. Before I start, I just would like to give you a hint note about University of Technology Malaysia.
Professor Hisham, I'm sorry. Can
Yes.
you share can you share the slides?
You cannot see the slides? I shared already.
أنا ما شفتهاش. حد شايفها يا جماعة؟
It's already
Yes,
there.
yes,
It's already
it's
there.
shared, Doctor. It's okay. Everything is okay, Doctor. Share. Maybe there is a problem with you.
It is already there. Shared already.
It's already shared.
Okay. UTM is currently 153 in the world. We are Premium Engineering University in Malaysia. And we have 3 campuses. The main campus in Johor Bahru, next door to Singapore. And the other campus in Kuala Lumpur. And the other newest campus is in Bago, which is education hub for Malaysia. And actually my job is between Johor Bahru and Bago, whereas I split the week between both, where I'm professor in chemical engineering in the Faculty of Chemical Engineering, and also director of the Innovation Center in Bago. If you're going to ask me where I am now, I'm not in these 3 places because I just arrived today morning to Jakarta. Therefore, I'm happy to be with you today, and I was worried about the internet, but everything's fine here in the hotel. Okay, when you're talking about mushrooms came with ancient cultures. Reported more than 9,000 years back, 9,000 years BCE. It means almost 11,000 years so far. They have some reports about mushrooms in the tribes on the North African caves. They find some drawing to say about mushrooms. The mushroom history, I would like to say, in 2 parallel big civilizations. Chinese culture and Egyptian culture. They start to value the mushrooms in their life as a food and as a medicine. But I don't know what happened for Egyptian culture. It's not continued with us until today. But for Chinese culture, this is very precious component for their nutraceuticals, cosmeceutical market. And this is a big business for them, the mushrooms. And they are right to tell what the mushroom is about. It's without leaves, without buds, without flowers yet. They form a fruit as a food, as a tonic, as a medicine. The entire creation is precious. Long time ago, they think it's a plant. But I think, as everyone knows, mushrooms is a fungus, macrofungi. In addition to that, in Roman ancient culture and Maya mushroom stones, all showing that mushrooms is deep in the history. What's the mushrooms? We find the different types of mushrooms everywhere. The button mushrooms, the straw mushrooms, the wood air mushrooms, shiitake, oyster mushrooms. Even you find mushrooms in the cartoons, which is the smurf, which is when I was a kid, I love it. And until now, by the way, I love it. This is from Stuttgart, Germany, this comic structure about the mushrooms. Then the kids become aware what's the mushroom. The world mushroom market, if you're talking about the mushroom market, is huge. When you're talking about medicinal mushroom market, is the second after edible, but not far away from that. In Egypt and Arab country, we think only about edible mushrooms. And I would like just to tell you, it's when I asked some friends, they said edible and non-edible mushrooms. I told them, no, all mushrooms are edible at least once. If you're going to eat again, it means it was not toxic. But you can eat mushroom, any mushroom you can eat anyway. But I mean, this is the way now with the business of about 63 billions business. It's a booming business. Where are we in this business? World mushroom market is led usually by lentinula, which is the shiitake, lentinula odidis, Pleurotus ostreatus, the oyster mushroom, auricularia, agaricus, flammulina, and volvurella, and others. But there are some strains or some mushrooms we can cultivate in our area and some mushrooms cannot. By the way, if you look also for the cultivated mushrooms in China, China is leading the world market in terms of production and consumption. Then after that, Southeast Asia, we are a growing market as well for Odidis and EU, America, and other countries. If you look for the distribution, again, this just remind you again to say, what's the leading mushroom? Lentinula and Pleurotus. Sorry. If you look for the wild mushroom about Pleurotus, this slide, this photo is actually from City of Scientific Research, where this is back to the year, I think 2003 or 2004, where we had the first greenhouse there to cultivate mushrooms. And the world market, as I mentioned, is led by China for most of the mushroom, but for Pleurotus is more because they have 87%. The world mushroom market, maybe you find in my talk, I love to link between the science and business because I love to do translational research since a long time. I work with industry and industrialization of the research. And therefore, when we go to the lab or we start a scientific research topic, I love to see the business impact. And if you look for the world mushroom market, for the same mushrooms, they have different pricing in different country, but they have a normal average price. And like example, Agaricus bispora, this is usually range between 1.2, 1.1 to 3. Some extreme market, like we find it's in Israel and other country, they have higher pricing. And by the way, this is just from statistical report. I don't mention specific country here or there, but this is based on the international reporting. Lentinula odidis, it's you find its higher value a little bit, and then If you go for sun mushrooms, it's about $13 per kg. But if you look further, if I need to say the top mushrooms, cordyceps. The cordyceps current pricing in the market, it's the price, oh, sorry. The current price of the market is more than 50,000 USD. This type of mushroom, cordyceps. Then we have the white truffles, then have Mitsutake, black truffles, morels, and others. It means don't underestimate the value of mushrooms. When the people say, oh, mushrooms is cheap, no, mushroom is not cheap at all. Mushroom is precious and expensive product, as I'm going to show to you in the upcoming slides. If you look for, in summary, just you said about the mushroom market, China is the main producer and consumer of the cultivated edible mushrooms, with 87% of the total production. Lentinus udidus, which is the most wanted now, with 20%. Pullurotus lentinula, auricularia agaricus. The world average consumption, 5 kg per capita per year. This is world average, even though some countries, they don't consume at all. But if you ask ourselves in Egypt, among the audience, how many kg have you consumed last year? Some of us, maybe they didn't consume even 1 gram. But if you ask this question in China, it will be totally different because it's in every dish. This is one of the lovely publication for me in my journey, even though this is 14 years old, but it was in trend in biotechnology when we were trying to investigate the medicinal mushrooms. And the figure, by the way, didn't change too much throughout the year. Agaricus, cordyceps, which is the caterpillar mushrooms, and Ganoderma, Grifola, Frondosa, and different mushrooms. They are the top 10 medicinal mushrooms of high value we usually consider when we're talking about the medicinal mushroom. Everyone talking about antioxidants of oyster mushrooms. Even of natural products, I hear always from people, oh, antioxidant, antioxidant, antioxidant. If you ask me personally, antioxidants no more value nowadays. Because if you just eat, if you're just coffee drinker like me, having 3 cups a day, I don't need any antioxidants more anyway. Coffee is the strongest antioxidants known. And if you eat grape, if you eat pomegranate, all those are very strong antioxidants. Therefore, when people just talking about antioxidants of oyster mushrooms, or they take some tablet or supplement antioxidant, for me, it's not high value, but they are more better value. Therefore, when the people say to me, oh, oyster mushrooms or mushroom have antioxidant, they said, it's common lie. It's almost like to say they have some protein. But, which is I write here in the right side, the low value compound or bioactives, hydrolysis enzymes, proteins, and antioxidants. But what we are looking for in mushrooms, antibacterial, antifungal activity, because they are active even against multi-drug resistant microorganisms. Antiviral, anti-tumor, immunomodulators, which is most of the mushrooms, because the presence of beta-glucans, which is I'm going to go in depth now, immunosuppressive and anti-allergic, hypoglycemic, anti-inflammatory, and hepatoprotective. Those are the molecules I'm looking for. These are some products in the market with quercetin, with lentinan, schizophyllan, polyuran, cordyceps. This is very common in Southeast Asian markets and world market, even if you go to Europe, Southeast Asia, China. This is one of the main components of the drugstore. The mushroom biobusiness focus for high value. Discovery of new anti-cancer, immunostimulants, or other bioactive compounds. Production and bioprocess development and scaling up of the bioactive polysaccharide production in submerged culture up to the bioreactor level for both known and new compounds, which is production package for pharma industry. Optimization and the isolations of the extraction procedures, which is also very important because downstream, we are also looking for that. Maybe these slides I just put to catch your eyes. when talking about the mushroom cosmetic market, which is the people don't know about it. This is a huge market. And I consider this is the pearl of mushroom business nowadays for mushroom cosmetics. Even if you Google after this lecture and see how big is this mushroom cosmetics, what's the mushroom cosmetic products, you will be surprised about that. There are two components of the cosmetic market. People forgot about it, and this is now booming. Mushroom cosmetic and probiotic cosmetics. And both are of high interest for me, actually, and my group, and in collaboration with many industrial partners. The market size can reach 1.9 billion USD in 2023, with accumulated growth rate 4.4%, which is considered very high. If you look for this bioactive prepared mushroom utilized in cosmetic production, they have functional properties: anti-tyrosinase, anti-inflammatory, antimicrobial activity, and anti-collagenase to protect our collagen. Okay? Antioxidative is a normal for me. But the other properties, no other bioactive compounds known to have this one. These are some of the mushroom cosmetics. If you find in Amazon or Googling, you will find the average retail price between 70 to 120 USD. Can you imagine if you, in my opinion, as one who work with industry, I would like just to tell you, the production cost for all those products, the best of the best, will never reach $10 per bottle. But the rest is the added value, is the R&D. Because this is a totally new product, then the profit margin is huge. I'm not talking about mushrooms. You produce in the farm, at the production cost for you, maybe 20 pounds, and then you sell in the market with 40 pounds or 30 pounds. It's not bring a business. This is not bad. I don't say stop it, but this is not the one you can evaluate mushroom to say, okay, mushroom is not profitable business. No, you go in the food market. But if you go to another market, like a cosmeceutical market, nutraceutical market, or pharmaceutical market, it's another story. The oyster mushroom, which is, I just described to you today, two platforms, which is I developed throughout many years in Egypt and in Malaysia. And we did some technology licensing to South Africa for this technology for oyster mushroom and cordyceps. The oyster mushrooms, they claim to the market, they said low sodium, fat-free, and low calories. This is to speak with the public, to make them attractive, to compare with other food. But we did a different cultivation system for the mushroom. We have the greenhouse cultivation system. We have submerged cultivation system, shake flask and stirred tank reactor, and air sparging system, it's covered by 2 patents and 1 trade secret, which is another cultivation system, submerged cultivation. This is, I would like just to share with you. This is the first greenhouse I established with Professor Ayman Daba, Professor Mahdi Milawi, and other friends. This is back to the year 2003. Institute of Scientific Research. You see how it was difficult to cultivate mushroom in Egypt? Because in summer, this is very hot. You need ventilation, and also you need humidity. It's dry. Then you need also atomizer. In winter, it's very cold for the mushroom. But when I came to Malaysia here, I would like to say the best business you can run in Malaysia is the greenhouse of mushrooms. Why? Because Malaysia, Indonesia, they don't have 4 seasons. They have throughout the year temperature 25 plus minus 4. It means we are living in the big four. We find it's a lot of investments, even Chinese investment nowadays, come to the regions to have a greenhouse. But when you go for greenhouse advantages, easy cultivation system with low investment, simple control, temperature, humidity, scalable process, the yield of purification is very low. This is sometimes they consider as disadvantage. Some people, they say it's as a food, it's fine. But the main disadvantage for me, many wild mushrooms are non-cultivable in a greenhouse. Many purification steps are required to obtain the final products in pure form, not completely sterile system, according to CGMP requirements for drug manufacturer. I cannot produce mushrooms in a greenhouse and I go to biopharmaceutical market because CGMP requirement. But in stirred tank reactors or submerged cultivation system, it's a complete sterile manufacturing according to CGMP, highly controlled production process, controllable and optimizable, short cultivation time, 15 to 20 days, and the feed batch only 20 to 23 days, scalable process, highly purified products, product formed are as extracellular products, but disadvantage here, feasibility, cost of production, system productivity, and production scale. This is just, I would like to share with you, just published last year, to make it describe a little bit for this business. I would like to say in mushroom business, there's something called gray business and red business. Gray business, it's you are not a food and not a drug. You are supplement. In this case, if you go through CGMP production, extraction, purification, structure elucidation, in vitro testing, more than enough. No need to go up to clinical trials. Like most of the mushroom products in the drugstore, they never say treat off. No, treat off, it means they did clinical trials. But they say enhance immune system, regulate blood sugars. All those can be done in vitro and proved and validated, but not go to the reach of the drug. Why? Because to make mushroom as a drug, they need a lot of investment and a lot of time. Therefore, most of the business go in gray business, in my opinion, in the mushrooms and probiotics and all those things, which is always forgot in Egypt. Everyone say anti-cancer drug. I need to produce anti-cancer, anti-what? To reach this stage, you cannot go through this one. To have a validated FDA drug, you need $100 million in your back to support. But start slowly, at least to have nutraceutical products to the market. Push to the nutraceutical, cosmeceutical products to the market. And later on, maybe you can find a big investor to help you on this, which is mostly usually it's big biopharma company. I'm talking now about the first molecule which is produced for many years, which is polyuran, which is alkali insoluble polysaccharides composed of beta-galactose and D-glucose with a ratio 2:1, triple helix conformations, molecular weight 762, but sometimes once you reach 300 kilodalton, still can consider as active molecules as immunomodulators. Bioprocess development, which is my field. When the people say to me, my friends ask me, okay, what you are doing actually? My first degree is microbiology. I'm a microbiologist, chemistry. And then my first master is related to microbiology, PhD, industrial biotechnology. Then my second master is a biotechnology business. You may find different group work with me from fertilizer, biological control, mushrooms, recombinant protein production, mammalian cells sometimes. But the people ask me what you are doing. They said, I just squeeze cells to make money. Like my professor in Germany. They're following the same as his concept. Then when you go for the platform here, I need to adapt mushroom to grow in agar plate culture. Then submerged cultivation in semi-defined, completely defined medium, characterization of the fungal biopellet formation, development morphophysiological model, and then scaling up of the process. This is called process know-how development, part 1. This just I would like to share with you. This is historical logo. Come back, I think, to the year 2001. Or yeah, it's almost 2001, 2002. When I come back from Germany and work in City of Scientific Research with my friends, Professor Yasser Rifat, we start together, and Prof. Salih Matar and Prof. Osama, Allah yarhamu. We started together to have bioprocess development department. And one day I received a visit from big professor He's a microbiologist. And he said to me, what you are doing in this department? Are you isolating strain? I said, no. Okay, are you characterize protein? I said, no. Are you use the microbe for bioremediation? I said, no. Then what you are doing here? I just moved from shake flask to bioreactors of different size. Then it came in my mind to ask him very simple question. How big is your family, professor? I said, me and my wife and 2 kids. Then I told him, okay, if you call now your wife, I said, I invited 10,000 guests. Can you cook rice for us? He said to me, she will say, I need a chef. I said, exactly, this is my job. How to produce rice for 10,000 persons, not watery, not burn, and taste well. This is the bioprocess development, which have a lot of bioprocess engineering variables. At that time, I decided to have a logo for the department, and I designed this logo, Bioprocess Development Department. The middle here is a bioreactor, BID, Bioprocess Development, the shaft, and we transfer from DNA to inside the reactor. It means from a scientific value to a business value. Then, I love actually this logo because it summarizes the bioprocess. At that time, I don't know how it's this come in my mind, but I like it until now. This determination of process bottlenecks, and then bioprocess optimization for the lab scale. And then we go for different strategy up to the scaling up of the process. And then we go for downstream processing for separation, isolation, purification. And then I optimize further this process with my students, Parisa Maftoon. She's Iranian and she was working with me. She is now in UK. She did the master with me for mushrooms. And one of the most important things is the growth rate. Mushroom is slow grower, but each day means money. And we developed this process in UTM for having fast growing media design for mushrooms. This is in short time in a petri dish level. Then we did after that medium screening for her thesis. And this is normal platform, as everyone knows. We screen different media. We have a production of the exopolysaccharide, about 2 grams per liter. And then we study the morphology of Pleurotus ostreatus in the shake flask, because morphology play a very critical factor in the process, because of this is for fungus in general. Once the morphology change, the whole process parameters change and the yield will be changed. And then we optimize the medium, different factors, carbon source, nitrogen source, concentrations as a conventional approach using fat, 1 factor of fat, that's 1 factor at a time. And then we did statistical optimization using RSM. And then we optimize, we start from 2 grams per liter and we reach 3.65 grams per liter. But still, it's not industry feasible. It means this is for the exopolysaccharide, polyram. This just, I would like to share with you where the story starts again. This was some photo from City of Scientific Research at that time when we was doing some research in the petri dish to bioreactor. And this is just I would like to share with you. The people not in the process, they think, as the professor said to me, just scaling up. They think I just go from small flask to bigger flask. It's not scaling up. Whatever, still using glassware flask, it's not scaling up at all. But the most important things, you need to find an answer for the investor when they ask you, how much does it cost to produce 1 kg or 1 gram of your product? And this is the gap between academia and industry. This is the very big issue, because investor he need to calculate how much will be the profit. If you cannot find an answer for him, for this one, don't invite investor. We need to find the answer first. Then we studied in depth the mushroom biopellet. This study is run in Malaysia again with one of my students. And we studied the fungal biopellet based on a model which is I developed in my PhD. It was for Aspergillus niger, but it's also applied for mushrooms. We calculate the biopellets formations, and we usually find if you have a big pellet, non-productive. Production is less because only the outer part is exposed to oxygen, which is the layer W. And then They are active fraction and inactive fraction in your cultivation. Then we need not to exceed the biopellet not more than 400 micrometer, not to losing any biomass as non-productive. Then this is for medium optimization, and this study was run by my students. If I give you the name, you will surprise. He's a Malaysian, but his name is Muhammad Helmi Misri. Therefore, I say you are more Misri than me. Misr in Malay language is Egypt. Muhammad Helmi Johari Misri. Johari, we are in Johor state. If you say we have a family in Egypt, Johari. We are having a state in Malaysia called Johor. You see how close Malaysia and Egypt, even though it's not, distance is not a matter, okay? Therefore, I just shared with you this one. And I remember Muhammad Helmi, he developed this with me for optimized medium, for optimizing this process. And we reached 3.45 gram per liter. And then we designed the feed batch cultivation system to reach at that. This was in large scale fermenter, 150 liter fermenter in UTM. The biggest fermenter I had is 1,500 liter, but we go 150 liter only for this process. This share with you when you're talking about the biopellet. When talking about biopellet, they have different factors from literatures and even by experimental. We have strain-dependent factors, cultivation condition, and medium composition. But the most important factors for scaling is the shear stress, mixing. People say shear stress, it means just increase RPM. No. Changing the impeller diameter and design, more important than changing the RPM. RPM is cost. And for us in scaling up, I need to produce with the lowest RPM, not to consume a lot of energy. But I change the diameters and the shape of the impeller. This is the platform I would like to share with you. This is in situ scientific research. Where is the story begin? Again, I just would like to show the story is back to 2003 until a couple of years back, in continued between Malaysia and Egypt. And we start 15 liter, 70 liter scale in Egypt. The maximal scale was 300. When we make a process design, we did something unique design in a way. I just described this to you now. Okay, we start with the 15 liter bioreactor. We go, this is trial transfer, the yellow one. 70 liter bioreactor, I can harvest straightforward or I transfer to 300. I have media preparation tank in between if I need to do feed batch. And then the novelty in this process here, I take the cells and the supernatant include low molecular weight polysaccharide. I pump again as a medium. Why? Because of polyuran can tolerate heat. I can sterilize even. Therefore, I am not losing those low molecular weight compounds after ultrafiltration. I do ultrafiltration, more than 300 kilodalton is my targeted compound. Less than that, I'm not throw away. I recycled again in a medium preparation again to increase the yield. And we develop a process using lyophilization, not using solvent extraction. Then we optimize the extraction process and to make its economic extraction method completely green process. And this is the platform. After fermentation, centrifugation, I separate the biomass. Even the biomass itself, we convert to a product, by the way. I just would like to say, this was the best if you're going to use for pasta. And pasta, mayo, it's amazing. If you're using this is a very clean mushrooms, okay? And then we have ultrafiltration process, and then you go lyophilization, or you go through precipitation process. And multi-step washing use ethanol acetone. This is one platform, but this is a green technology, which is we developed and we patented this one to ultrafiltration and lyophilization. It was detected after that using IR and 13C NMR analysis. This is showing that it's linking mainly 1316 beta linkages, beta glucan. And this amazing work was done by Professor Mahdi Milawi. I cannot show a lot of slides because of the time constraints, but she shows that this is for it inhibit cancer cells for in vitro analysis. This is the polyuran. This is the first part. I would like just go the second part, which is, for me, was very interesting. Cordyceps for years. This is the mushroom I mentioned to you, if you remember, this type of mushroom, 50,000 USD per kg, because they have a cordycepin and another bioactive compounds. This is the most expensive mushrooms. What's the cordyceps? You will be surprised about this one. Cordyceps is a fungus, come from the head of dead insect. And it's known in Himalaya area, in the north of India. Even during this season of cordyceps, they close the school and make them go to collect cordyceps. A fungus comes from the head of the dead insect. Would you like to try? Therefore, where position cordyceps? Is this disgusting museum in Netherlands or healthy food? If the people knows that it's come from dead insect, it's still not acceptable. And for years, it was very difficult, no way to cultivate this type of cordyceps in the lab, non-cultivable mushrooms. But nowadays, I think in the last only 10 years, there are different groups, they cultivate cordyceps in the lab. And among this group is our group. In collaboration also with Shizuoka University, they have surface cultivation. We developed the submerged cultivation for scaling. The cordyceps are cultivated, collected, and applied as edible and medicinal mushrooms. They have umbrella-shaped. They have a macrofungus, and it's a macrofungus from fruiting bodies. Cordyceps is a class of Claviciptaceae and Ascomycetes. And this is the most important one, is the militaris, Cordyceps militaris. In addition to that, it was in the, I think if most of you have been to Germany or studied in Germany, the famous journal, famous newspaper, which is Frankfurter Allgemeine, they mentioned Das Viagra des Himalayas because they have some hormonal strength for men as well. But the cordyceps, most important things for us is immune system enhancer, anti-cancer properties. And it's widely used in Chinese medicine. Maintain healthy liver function, hepatoprotection, maintain healthy respiratory function, and enhance kidney function, lowering and maintaining cholesterol level, hormonal balance, and improve memory. Most of these have been proven in a lot of studies. We have top 10 cordyceps species, but the most important one for us is Cordyceps militaris, which is we develop a process, and they have different type of cordyceps you find in the market. Some of them are fake, by the way, and authentication of cordyceps is very important. And also, by the end of the day, as I mentioned to you, if you produce coming from insect, it's very difficult to go through as a CGMP product. You need to cultivate under sterile condition. The most important compound is cordycepin, which is low molecular weight compound beside the polysaccharide, which is known. But this is straightforward anti-cancer compound. This is actually, it's done through the PhD of my students, Muhammad Sultani. He's also Iranian, by the way. And we did a good job together to optimize this process in scaling up in the fermentation. We studied in the OFAT, we optimized the medium. We did a lot of study in the bioreactor level, batch, feed batch cultivation, statistical optimization, and we optimized a complete industrial platform to produce Cordyceps militaris. For this, in summary, from unoptimized, you see here the yield, the biomass 21 gram. But if you look for the product, cordycepin, only 148 milligram per liter. But for me, is more than enough. People say it's low, but if even produce 0.5 gram per liter, for me is fine. Because if I am going to cultivate in 1,000 liter bioreactor, it means I produce already half kg of pure polyuran. In this case, it's almost 20,000 USD. I'm not saying about retail price, or let's say go down to 15,000, I'm fine. Okay, then we optimize the process through unoptimized, then optimize through OFAT, and then statistical RSM. And then we go in the bioreactor cultivations, because before this one, we cannot say we optimize a process. I must go through a bioreactor. Then, even though we have here uncontrolled pH, we find uncontrolled pH giving better result, and up to 446 gram per liter. And then we develop a process, feed batch, and we can reach up to 800 milligram, which is considered very high. I'm close to 1 gram now. Then we reach this level. We are very happy that we reach this cultivation process of cordyceps, and especially this calculate here, product here, it means cordycepine. I'm not calculating the polysaccharides. Polysaccharides, it's another value, products as well. But I was focusing in cordycepine. In summary, screening programs of the wild mushrooms, I would like to say ignorant wealth. Still, we have a lot of mushrooms in Egyptian desert, in Malaysian jungle, in everywhere in the world. They say non-cultivable mushrooms. But nowadays, we optimize different process with a lot of tricks, a lot of new medium ingredient to cultivate most of the mushrooms. The other things when you say about toxic mushrooms or poisonous are amazing because they have a lot of bioactive compounds, can be used as anti-cancer, by the way. They are now a big trend to cultivate poisonous mushrooms, but those laboratories need to be controlled like biosafety level 3 lab to cultivate and produce some bioactive compounds, which is of high medicinal value. Cell banking of the rare mushrooms. Even just recently, I was in a discussion with some group in China, for Botox. I think people knows about Botox in plastic surgery and for cosmetics. This to say, even though people, microbiologists know Botox is a toxin, which is Clostridium botulinum. The same, they said they have discovered some bioactive compound. They act as a Botox, but less hazard than Botox come from poisonous mushrooms. But still, they do a lot of study about this one. Cell banking of the rare mushroom is necessary and it's doable because without cell banking, you're going to lose the strain. Greenhouse is not enough to judge the cultivation of the wild mushrooms. New cultivation systems and strategies are needed to produce the mushroom bioactives. Stirred tank bioreactor and air sparging cultivation system, which is I didn't show here actually. It's another totally different platform. Cost, I would like to say peanuts compared to stirred tank bioreactor, which is I developed in City of Scientific Research and patented this one back to 2006 actually, we patented this one. And I did also improvement of this system and licensed to South Africa. It was in the year 2019, just before COVID. And then we need to develop new cultivation system, economical for the mushrooms. Mushrooms as food is good for zero hunger. Everyone talk about, before I leave this to the last point. Mushroom is, people think, oh, high protein. For me, it's nothing. Soybean is higher in protein. Soybean price, you cannot compare to mushroom at all. This is the lowest protein source in terms of costing. Lowest cost of protein. And then, however, we need to think about the health supplement, pharmaceutical market, as well as high value business, cosmeceutical, pharmaceutical, nutraceutical market. These some of the publication in my journey. We had 2 patents, 1 trade secret, 10 book chapters, 17 papers, 24 conferences. And we have another industrial secrets to 2 companies. And with the big groups I work with, I honor to work with Dr. Daniel Joy Dylan, associate professor now, and Dr. Sultani, Rosalinda Malik, Marhuma Rosalinda Malik, Dr. Siti Zulaiha Hanafi, Mr. Saleh Ramli, Parisa Maftoon, Muhammad Helmi Misri, you see, and Zay Kse, this is also one of my students. Industrial partners from Malaysia, Harita Gogreen, And from South Africa, Sapuna Mycelium. And from Hong Kong, Dr. Ting Hu. From Lund University, Professor Rajni Hatikaul. And from Universitas Negeri Jakarta, Prof. Mokti, Prof. Dalia. And from Baghdad University, Mr. Ramzi Abdul Sahib. And from Basra University, Dr. Jafar. And from City of Scientific Research, where does the story begin? My brother, Professor Dr. Ayman Daba, Professor Dr. Mahdi Milawi. And now I collaborate recently for the second generation of development with Professor Dr. Nihal El-Deeb and Dr. Ahmad Atta. Engineer Salah Al-Sayed and Engineer Ahmad Ali, they are the one who work with me day and night in City of Scientific Research to develop this platform in the early days. Ahmad Siddiq and Islam Al-Badri, Abu Saud, and Ahmad and other in technical teams. National Research Center, Dr. Sayed, Professor Dr. Sayed Ahmad Sayed, and from King Saud University, Professor Dr. Muhammad Al-Wad'an. In this journey, we work with different types of mushroom, but mainly I was focusing on Pleurotus and Cordyceps, Shiitake, Ganoderma. We published with Universitas Negeri Jakarta and others. In conclusions, I love to say, I'm a Smurf guy. I love Smurf. I don't like Mickey Mouse too much, but I love Smurf more. Okay, Smurf loves mushrooms. If I love mushrooms, then if I love Smurf, then I should to love mushrooms. And this is my professor. I work my PhD in fungi. I said to work with the fungi, you need to be a funny guy. Why? Because of fungus make a lot of contamination. If you cultivate mushrooms, cultivation for 20 days, and in the day 18, contamination happened, you need to be patient. You need to still smile. I remember how many times my dear friend, you have Sultani or Parisa Maftoon, they used to come to cry in my room. I say, just have a cup of coffee and relax. You need to continue because the mushroom is the big story about mushrooms is you need to have, when you cultivate mushroom in a bioreactor, you need to have a very clean and sterile condition. With this, I would like to say thank you very much. This is part of my team for Innovation Center of Agri-Technology for Advanced Bioprocessing. And I would like to say thank you very much. I hope it was not boring for you to hear this lecture. And I try all my best to make it sound in a nice approach. And I hope you enjoy this one. And yeah, I'm looking also for collaboration and you come to visit us anytime in UTM. Thank you very much.
Thanks, Professor Hisham, for your very nice talk. I think now we can open the discussion. Anyone have any questions?
Yes.
Can I have one? Oops.
Okay.
Yeah.
After you, Doctor. Okay.
Is it okay? Can I speak now?
Yes, welcome.
Yeah, you can speak.
Professor Hisham, as usual, يعني حضرتك أتحفتنا.
Oh,
I remember
thank you
2 years ago, you spoke about a value of microorganisms from the sea, and was excellent. يعني دائما حضرتك you lead us with new ideas. حقيقة. My question is,
Professor, please can you introduce yourself?
Yes, I'm Ahmed Abdel Mawgood. I'm a professor of biotechnology at EJAS, Egypt Japan University of Science and Technology.
Welcome.
Allah يخليك, شكرا. Just 2 weeks ago, we make a survey about isolating whitewood fungi, but I isolated one mushroom from Wadi Natun. I just like to hear from you. Where to start? How to start from that? Also, my student is listening to you now. Thank you.
Okay, excellent. I mean, number one, if you isolate mushroom from desert, you need to be very careful to ensure that it's not poisonous, because of the spores could be inhaled easily. But anyway, I'm not scaring you, but okay.
Yeah, okay.
I need just to have gloves and everything need to be careful.
Yeah,
And then
we do that, yes.
it was able to cultivate on potato dextrose agar and the normal media in the lab,
Yes.
or it was
Yes,
not
it was.
possible?
Yes. Yes, it
Excellent.
was.
Then you are lucky for that. Then the second things we need to make identification. The good things in
Yeah.
mushroom, even with morphology, you can identify, giving you brief, I would like to say identification, we can see up to 80% based on morphology. And after that, you need to have molecular identification. Once you know that, you will know under which group. Is this safe or non-safe? Even if it's non-safe, but your lab need to be equipped with a biosafety level 3 laminar flow to work with it. If
Yes,
it's safe,
we do have.
then it's okay. I mean, if you are lucky, it's safe, then we go through that way. My approach is very simple. I look for the function before the molecule. I'm straightforward to the business. First, you need to cultivate to see, is they produce a polysaccharides? And is this functional polysaccharide can act as what? Enhance immune system. This can be done in vitro. Or antioxidants, I don't care too much about that. But enhance immune system, they have potential anti-cancer activity or not. They have prebiotic property or not. Because of this is the future industrial claim. You can tell about that. If have, then go deep. Okay, what's the molecule, how to isolate, what's the bioactive compounds here and there. Low molecular weight, high molecular weight. You need to consider both.
Okay, thank you. Thank you very much. I've been a bit.
You will come, Prof.
Professor Hisham, I have one comment about this questions. If he isolated mushroom from desert, this it mean it is a wild mushroom. Okay. And you in your presentation you said wild mushroom it is not recultivated. So how we can solve this problem?
No. The normal people say that, Prof. Hisham. I don't say all. Let's say if Prof. Ahmed have one of the known like oyster or whatever, this will be cultivated. Some other mushrooms, people say not cultivated. But we developed different cultivation media, not BDA nitrate, or no. We have another supplement we give to the culture media to make most of non-cultivable mushroom can be cultivated, like cordyceps. Cordyceps at the beginning, people say cannot be cultivated. More than 15 years back or 10 years back, no single papers about cultivation of cordyceps in the lab. They just isolation of bioactive compound from the wild. But nowadays, you find a lot of papers related to cultivation. But I would like just to share with you something. Most of the people, even in the paper, they don't reveal completely the medium composition.
Yeah. So I will ask you about this later. Any other questions?
We go to the private box then.
Yes,
Any
Dr.
other questions?
Yes. Thank you very much, Professor Dr. Hisham Al-Anshasi, for this informative presentation. Actually, even me, away from the specialty, I'm a pharmacist, but really I got many information about the subject. I hoped before to get this information, of course. I have be patient for receiving my question because as I said, I'm away from the specialty. So be patient, as you said at the end of your conclusion about the mushroom. We have to be all funny guys and be patient for them. Number one, Doctor, my information about mushroom, not all are safe. There is an editable and the other no. This is my study before. And this is why even I find out more mushroom in my own country in Guinea, Egypt. But I afraid this is safe or not. But you said at the beginning of the presentation, no, all editable and the toxicity may be low percent or not so far I have to care with. It's okay. I have to confirm this information also from you, Doctor.
Okay, thank you very much. I would like to answer your question in two ways. The first, as a pharmacist, you are not far away. You are in the business.
Thank you. I will ask
Without
another question.
pharmacists,
I will ask another question for this business. But
no,
this is the
no,
first one.
okay.
This is why this is my
Without
spirit.
pharmacists, we cannot go further. And by the way, today I think we have with us Prof. Rania Abu Zahra. She is the Dean of Faculty Pharmacy, Damanhur University. Okay?
Okay.
And without pharmacists, we cannot go. We need pharmacists in this one. The second one, maybe I did the joke in wrong way about the toxicity.
Thank you, Dr. Hisham. Thank you so much, Dr. Hisham. I'm here with you.
Thank you, Professor. It was what I say. It was a joke. I said all mushrooms are edible.
Yes.
At least once. If you eat again, it's non-toxic.
Yeah. Maybe I got.
You got it.
Yes.
I mean, I don't say. Therefore, when the people classify mushrooms, said, okay, toxic and non-toxic, not edible and non-edible. Once you have a mouth, we can eat everything.
Yes. I got it now. So I have to clear. Yes, thank you. I got it.
Okay.
Yes. Another question about my specialty as a business pharmacist. So in the figure, in the presentation, you divided two types of business. I target or I intend for a red business. You said if you want to get the pharmaceutical benefits from the mushroom, different types of mushroom, so we have to make an effort to get what we want to sell or what we want to distribute. But when I saw another presentation, the active ingredients which present in the slide already reported.
Yes.
So we need just extraction and purification, and I think this is at industrial level will be easy. So why you intended to put in a red business, as you say, Doctor? This is easy.
I would like to tell you this. My answer to the professor who came to my department, bioprocess engineering. Our just producing, scaling up. No. Production is not easy at all. Scaling up of process is bioprocess engineering. Is a completely different field. If you produce in a petri dish or shake flask, it doesn't mean you produced at all. You need to produce in a scale. How to produce in a scale? There is a lot of bioprocessing parameters. There's a lot of engineering variables. It's a lot of design, a lot of theories. Let's say if you produce in a small scale 2 grams per liter, but when we scale, we can produce up to 50 grams per liter. Then I am more economic.
So
therefore, this is the big. I love your question because this is the big gap we have in Egypt. The bioprocess engineering and the pilot plant, how we go from the lab to the industry. So even though we have many pilot plants in Egypt now, but people need to use.
Just need cooperation, but between different laboratories, different specialties, like consortium.
I just would like to say Egyptian Academy of Science and Technology, which is sponsor these wonderful programs. They built already 3 pilot plants in bioprocessing, in City of Scientific Research, in National Research Center at Mansoura University. And I was honored that I was part of this story and I did the training for the 3. In addition to that, we have now at Sadat University, they have another pilot plant.
Good.
They have also fermentation unit at Al-Azhar University, which is also, I helped on that long time ago. They have also another unit at Faculty of Pharmacy at Cairo University. It means facilities are there.
Yes, I got it. Yes, good done. And I'm sorry, the last question, maybe I take a lot of time for for the other audience about because now we are with the specialty of the subject. So I would like this is a common question. As you say, the mushroom containing many ingredients, many nutrients. What is the proper methods for done of cooking? Well done or half done to get to get all the ingredients from the mushroom? What is the proper way for cooking to be well
No,
done or half done? Because
okay, I tell you what.
all we use mushroom in our house. This is easily for us here
Okay.
in Saudi Arabia also.
Okay, I tell you what, I'm not expert in nutrition, but I give you a very simple example. If you talk about oyster, the polysaccharide, even if you cook up to 200 Celsius,
yes.
autoclaving, still stable. Protein gone. Okay, forgot about the protein, forgot about the enzymes. But the polysaccharide, which is immune modulators, very, very stable. It means it's depend on what's your targeting molecule. Cordycepin is very stable.
Good. What
It's
about
depend on
steroids? There is the steroids, the estrogen, and other cholesterols. Many steroidal nuclear moiety, steroidal moiety member there. What about it? Will be bear the
actually, I didn't study this one more, but I study more in depth about the polysaccharide and low molecular weight functional immunomodulators. Like example, there is also another very attractive molecules, which is fungal immunomodulators proteins, which is now the trend now. But I start this research for a time being in collaboration with other institutes.
Thank you, Doctor. Thank you. I understand. Thank you. Thank you.
There is another question. I think maybe Dr. Huda Al-Gharabawi. Here is her hand.
Okay, yes, thank
You
you.
have question?
Yes, thank you.
Okay.
Thank you so much, Dr. Hisham. It's a very interesting lecture and very joyful to have this knowledge from you. Really, I'm associate professor of microbiology and spent about 18 years in studying of mushroom in my studies in university and postdoc. And it's very
Which university?
Damietta University Faculty of Science. Yes, and it's a very interesting field. But I already have lots of information from you, and I really have a few questions about the lecture. You already talk about bioactive compound from cultivated mushroom, then wild mushroom, and we already know that wild mushroom already have more, I think, more efficient bioactive compound because it's used in survive in the environment. Do you think cultivated mushroom will have the same efficiency like wild mushroom? This is the first question. And the second question is you speak about submerged fermentation. Again, it's natural cultivation. I think for the mycelium or bioactive compound, the same question, will it have the same bioactive efficiency or it will be more, or there is some differences in the bioactivity for the product? And the third question is the red business, which you are talking about, and you already suggested that we should continue to the end of the pyramid to have accurate results. And we usually continue until the in vivo, in vitro, and we sometimes go for in vivo testing like cell line and some animal like rats and so on. Are they enough? Or we should continue for clinical trials because you know that it's sometimes restricted and we have ethics and so on. And thanks a lot again.
Okay, thank you very much, Doctor. Actually, you raise up a very important things. When we're talking about collection of the wild mushrooms, yes, some wild mushrooms, they have more bioactive compounds. But I talk about the business sustainability. I cannot rely on the wild mushroom to start a business. To do a papers or publications, yes, but I need to have a sustainable resource. Therefore, for sustainability of the business, you need to cultivate in the lab. I totally agree with you because of sometimes some compounds need to be in fruiting body, not in the mycelium. Okay, but what's different between fruiting body and mycelium is the aeration. Exposed more, the gels is more exposed to oxygen. Therefore, sometimes you will play in this process with the oxygen level to induce some bioactive compounds. It means, but even though if you say to me the yield of the wild mushrooms could be 20% higher than the submerged. Now I go for submerged anyway, because this is more sustainable. This is in a business point of view. When we go for the permits I showed to you, I means here, most of the time, the researchers look to have a drug. For me, don't waste your life to have a drug. Because if you are lucky, you have enough money, the one who's going to celebrate this drug is your kids, not you. Because clinical trials alone, it takes 10 years. Therefore, the way to development is not only for mushrooms, even for probiotics, even for many compounds. I say to the people I work with and my partner, just have intermediate products in the journey. It means I know polyuran, they have some functional property immune enhancer. Then I produce food supplement. With the company in South Africa, which is when I licensed to them technology for polyuran, they go for new products. When we sit together to develop a product, maybe you will surprise. Mineral water supplemented with a polyuran for the kids in winter to enhance immune system. And this is the best buy. They have pet feed for cats and dogs supplemented with mushroom to enhance immune system. They have the third product is mushroom cosmetics. They didn't go further as anti-cancer. No. They just make a lot of money through this. And in this case, could be a pharmaceutical company who have the capacity, they have the capital, they have the platform, they know how to register the drug, they know everything. They will come to you to say, okay, I will complete the journey. I have the resources, I have the money, I take it on my shoulder. But you're going to have a reputation in the market. Polyuran is a molecule, people know about it, it's good in cosmetics, it's good in enhanced immune system. Then it will be easy for them to complete the journey with you. But we as a researcher, no way to complete the journey. This is just from my experience. Is this okay, Prof?
Dr. Ahmed?
Yes, it's okay. Thank you a lot, Dr. Hisham.
Thank you.
Okay, just a minute. Please, Professor Ahmed Zohari. Please mute all the phone.
Okay, it's all muted now.
Okay. Yeah. We have also Dr. Ahmed Haddad. I think he has question because he raised his hand. Dr. Ahmed?
Hello, everybody. First of all, thank you, Professor Hisham, for this interesting presentation and lecture. We got a lot of information. Actually, I have a small question. You mentioned scaling up bioprocess engineering and bioprocess development. What about solid state fermentation? You mentioned bioreactors. You mentioned submerged fermentation. What about solid state fermentation? Because I get, if we are going with mushroom cultivation, one of the best way, I mean, traditional method to produce mushroom, even for food. For example, oyster mushroom, we cultivate it on wheat straw or rice straw, whatever. And it's quite cheap, and I think it's more productive. You can produce kilograms of mushrooms. So what about solid state fermentation and the way to develop it and to scale it up? Thank you very much. If you could shed some light about that. That's
Thank you very much,
question.
Prof. Ahmed, my friends since 25 years. And you was also part of the story. Thank you, Ahmed. Very happy to see you
Thanks.
today.
Thank you.
Okay, when you're talking about greenhouse or the solid state, this is the most suitable platform for food industry. But when you're talking about to produce a bioactive molecule, we prefer submerged. Shorter production time. You can play a lot with the medium components to enhance or to induce the production of some compounds. The third things, they produce as extracellular. I remember when we started the platform with Professor Ayman Daba, he used the solid state. Solid state, to produce the bioactive compound, you need to crush, heat, autoclave. It's a long process. But when we produce in submerged, we were able to produce as extracellular products, polysaccharides or cordycepin or whatever. I don't need to have many steps in downstream. This is the advantage. Okay, usually each system have pro and con, as I show in my lecture, but it's depend on what's your targeted application. If you're talking about food, yes, totally agree with you, solid state. But if you're talking about bioactive molecule to use as a nutraceutics or cosmeceutics, usually we prefer to have submerged, especially you can run under full sterile condition in compliance with the CGMP.
Okay, thank you. I think it's enough answer for my first question. Second thing is I got it actually from your presentation and some questions of the other audience. So I guess we should look, or we should shed some light about domestication of wild type mushrooms. Yeah, actually, I have a dream since long time ago to make like mushroom collection center in Egypt, so that we can isolate wild type mushrooms, and then we can, or try to domesticate it, to isolate it already in the lab. Actually, I succeed to do that in some strains, but I'm not sure whether it's enough or not. I guess we should go together with or cooperate with other colleagues that have interest to do that.
I totally agree with you, Prof. Ahmed.
Thank you very much, Dr. Hisham.
I totally agree. The main drawback was usually non-cultivable one. But nowadays, we have a lot of medium formulation with increased knowledge. Even truffles we can cultivate. It's not only mushrooms. Okay, some still problematic. I cannot say 100%, but large number of the wild mushroom. The desert mushroom is ignorant mine for Egypt. I would like to say that. Countries like Tunisia, Bahrain, Algeria, they did a lot for desert mushrooms, but they don't have the collection. After publishing the paper, it's disappeared. They cannot bring it back. This is the problem. But your idea about National Center for Mushroom Cultivation, this could be a very good idea. Thank you very much.
Thank you, Dr. Hisham.
We have another question from Dr. Nisreen. Hello?
Hello.
Nisreen?
Hello. Yes, I'm Nisreen from Tunisia. It was very insightful, and thanks a lot for the idea that is just mentioned about the Center for collecting wild mushrooms. Because me too, I'm intending to do that. I started that because I'm an agro-business engineer from Tunisia, and I'm doing research on that. For now, I did some collection of some wild
can you give the
mushrooms
full
here in
name,
Tunisia.
Nisreen?
I'm from Tunisia.
No, I know.
Yeah.
Nisreen, what's the family name?
What is your...
What? Sorry?
Your full name. Nisreen?
Nisreen Tamerhi.
Okay. Which university?
I'm University of Carthage.
Carthage. Okay.
Yeah, I'm at Research Center of Biotechnology of Tunisia here.
Okay.
I'm doing my startup at the same time on mushrooms, so it's business and research, at the same time. For now, I'm starting to collect some wild mushrooms like we find here in Tunisia. I found one in my garden. It's Ganoderma resinicium, and another one Ganoderma platinum, and I found also oyster mushroom. For oyster mushroom, I succeeded to make the fruit and body and all, but I'm afraid if I do a lot of transfer on media, it will the yield will drop. So I'm asking how to maintain the yield. And for the resinicium mushroom, I succeeded to make the spawn, but every time I add something to the dust wood, there is contamination. I couldn't make the fruit and body. Also, another question I would like to ask is about button mushroom. For button mushroom, I succeeded to do it on media, but every time I transfer it to the spawn to the wheat, it won't succeed. So, what is your advice for me for that? Thank you.
Okay, I would like to give you a general recipe. People, when you cultivate from BDA to BDA, potato dextrose agar, potato or Zapex, which is known for fungus, you lose some properties, for sure.
Yeah.
And therefore, what I recommend usually from time to time to put some lignocellulosic material even in the plate to still keep the cell produce hydrolases, because they survive on the soil based on hydrolases. Because if you don't give them normal food, I mean normal nutrients, then they lose some properties. And the other trick of the game, okay, I would like to tell you is okay. Try to think about sodium nitrate.
Sodium nitrate. What can I do with nitrate?
To add to your cultivation media, but not exceed 3 grams per liter. It's depend on the sensitivity of the mushroom. Some mushrooms are tolerate up to 3 grams per liter. Some, we just start with 0.5 grams per liter. This will enhance sporulation.
Okay, thank you. Thanks a lot.
Thank you.
Okay. And I would last, this year my professor is like, we're doing something on algae. We're going to add some algae on the mushroom. What is your advice for me for that? Do I add it to the media or the spawn or the fruit and body? What is the best? And also yes, another question. If like for the fruit and body, sometimes the conditions are not good here in the center because they're not specialized in mushrooms. So there isn't like enhancing the fruit and chamber and all. So what do you advise me to do? Do I measure the enzymes that the fruit and body can have, or see how do I measure the yield if I don't have the good environment like to measurement?
Okay, for the first one about addition of algae into cultivation media, I don't have experience on that. I mean, I didn't did that before. But we need to know why. Why? It's what's the special in the algae to be. Because there is another concept. I love usually a concept back to nature. Back to nature. Mushrooms never been to the sea.
Yes, so they don't need it.
Okay. This is just very simple concept.
Okay.
They not coexist together, not like lichens. If you said algae and fungi, this concept, but not fungus. Fungus have been used to be alone without algae. If you say lichens, yes, for sure.
And the mushroom is different.
Okay.
Yeah.
Thank you.
But could we have some? I don't know. Therefore, I said to you, I don't know. For the other one, we talk about fruiting bodies and the quality of fruiting bodies. For me, I don't use fruiting body too much in my work because my target is to produce bioactive molecule under sterile condition.
But those molecules are not in the mycelium; they are in the fruiting bodies.
Yeah. Therefore, I said to you, you need to change medium composition to induce the production of these. What's different between mycelium and fruiting bodies? Very simple. If you look for developmental biology approach, oxygen, then this is one of the components. The second component, which is I induce sporulation, because fruiting bodies, they have the basidiospore. Okay. Therefore, you need to create a condition to enhance sporulation. Therefore, I said to you, sodium nitrate. It's known that sodium nitrate enhances sporulation. Then it can start or switch on sporulation cycle to fasten the growth and induce some molecules.
Okay. Thank you a lot. It's very insightful.
Thank you.
We have another question from Professor Kamel Shakir.
السلام عليكم بروف هشام. السلام عليكم.
السلام ورحمة الله وبركاته.
Many thanks for you, Dr. Abdulatif, and also Professor Hisham, my brother and my friends. And my question could be come to the chemistry point, not to the bioprocesses and so on. Because you mentioned, Professor Hisham, for a lot of compound or many compounds from mushroom, and mainly you are focusing on polysaccharides. And I'm very interested to for structure determination or structure elucidation of the polysaccharide moiety. So the best way to identify the structure and the connectivity between the sugar unit, for example. Thanks a lot. That's my question.
I will say in Arabic first, Habibi, Dr. Kamil. He's my close friend more than 30 years. And I remember last year we had a nice breakfast in Bosphorus in Istanbul. Okay. And okay, come to the question. I would like to say, if you look for this polysaccharide identification, we usually go for different techniques. I think you are the expert on that. You are the chemist guy. And nowadays we look for molecular weight diversity of the compounds, like capillary electrophoresis, to know that. But the other approach I would like to say to you, for me in my research, I am not a guy who investigate for the discovery of a new compound. Maybe I'm looking for mainly to industrialize the process of the production. Therefore, when we collaborate together, it will be excellent, Prof. Kamil.
Thanks a lot. I thought also maybe one of your group, of course, specialized in the structure determination. So really, maybe the facility could be not easy for structure determination for such a molecule. So I'm interested to know.
This is because it's not about equipment, Prof. Kamil. I think you know.
Yeah, of course,
It's about the expert.
Tamam.
Who read the chart. It's not how to produce the chart.
Okay, my Habibi, okay. My Habibi, okay.
Okay.
هو حبيبنا كلنا ما شاء الله.
الله
ربنا يخليك
اكيد
بروف.
اكيد بروف هشام، اكيد بروف عبد اللطيف، ربنا يوفقك.
ربنا يبارك فيك. Is there any other questions? أعتقد ما فيش تاني ولا إيه؟
احنا ممكن لو ما فيش أسئلة
دكتور هايدي رضا. دكتور هايدي رضا has questions. دكتور هشام. دكتور هايدي رضا has question. Okay, Dr. Heidi.
Thank you, Dr. Hisham, for this inspiring lecture. I'm Heidi Reda, third-year biotechnology student. My question is, what are
which university?
Your university.
بني سويف.
بني سويف.
ده من عندي ديها.
أهلا بحضرتك يا دكتور.
لا انت عارف بروف هشام، انا مواليد الفاشن، فعشان كده يعني ايه
انا كمان.
ما شاء الله.
Can I speak Arabic?
Okay, please go ahead.
تمام، انا طالبة في بايوتكنولوجي سنة ثالثة. كنت حابة اعرف من حضرتك واستغل
Please tell it in English because we have a couple of
ده
اوكي.
بايوتكنولوجي عندنا في زراعة ولا فين؟
يا دكتور.
دي عندي يا دكتور هشام، دي في كلية زراعة، عندي في برنامج بايوتكنولوجي.
ما شاء الله. Yes, please. What's your question?
ممكن.
تمام. My question is, what are the most critical laboratory and bioengineering skills we should focus on to bridge the gap between academic research on mushrooms and the industrial scale-up, and how we can start developing ourselves in this field?
This is a very nice and tricky question. This is the future. I would like to say that. We should to have
تتكلم عربي.
we should to have some courses I recommend for the basic of bioprocess engineering in the biotechnology curriculum. Then They know what's next, how to go. It's not just to go from shake flask to bioreactor. Bioreactor is a big science, or cultivation vessels. How to design, what's the material, how to do the process, how to optimize a process. I think in my lecture you find a lot of equations. And we work with a lot of equations to optimizing the process. Therefore, I recommend here, Prof. Hisham, to have integrate some of the curriculum basic of bioprocess engineering for the biotechnology students.
Inshallah.
Inshallah. And I am always there for any help, Prof.
Thank you.
Okay.
Okay.
Thank you.
Thank you.
If there is any other questions?
دكتور هشام دكتور عبد اللطيف هل ممكن can we wrap up because we don't want a lengthy lecture.
Okay.
So this is because this will be uploaded on the YouTube. So we don't want it to be a lengthy lecture.
Sorry, Dr. Ahmad, can we have the contact detail of Dr. Hisham, please?
You can contact Dr. Abdul Latif Hisham or Dr. Hisham, and we will upload this lecture on YouTube, my YouTube, and on the Facebook. The same
Okay,
places that
I will.
you see.
It's
saw
easy
the,
also. I will, if I
yeah.
find the chat box here, I can write my email.
Yes.
If anyone would like to communicate, I don't find here the chat box, but my email is very simple. Hisham@utm.my. HESHAM.
You can write it on the chat.
@utm.my. I don't find where is the chat box here.
Chat. I'll just write something in the chat.
Doctor, at the bottom, after participants chat, after react chat, at the bottom of the screen.
I have more. I have host tools, share. Oh, chat. Okay.
Chat,
Okay.
yes. You
Got
have
it.
to press just, okay.
Hisham. Okay. Okay.
And also, please, I would like to ask all of you to write your name and your affiliation. This is important for to get your certificate. Okay?
Okay, دكتور عبد اللطيف إن شاء الله جزاكم الله خير.
Yeah, inshallah, bismillah. We have another question from Karim Ahmed.
First of all, thank you very much, Professor Hisham. This is just an insightful meeting, and I have really excited for this information. Thank you so much. I'm an undergraduate student at MSA University, undergraduate biotechnology student.
Which university?
MSA University.
Yeah.
Last year, in third year, I have researched on concept of industrial project for concept of lignin degradation in corn residues by Trematus, followed by Pleurotus cultivation for nutrient enriched animal feed. Actually, this is just an innovative, creative title, but I'm not deeply researched on this concept. But the main aim of this concept is to make low-cost, efficient nutrient feeding for ruminants. Okay, so I have just a question regarding to Pleurotus. For my title, I have used lignin degradation using Trematus, and I have used Pleurotus for cultivation to enrich these ruminants as a cost-effective method for nutrient feeding for ruminants. So I wonder which method is better. Should I use pre-treatment chemical method rather than Trematus and then Pleurotus, or is it better to use the lignin degradation using Trametis, which is a one fungi, and another type of fungi, which is Pleurotus for nutrient media. Thank you.
Okay, if you're having Trametis first, it's you are targeting lignin degrading, correct?
Yes, exactly. I have.
Lactases, especially lactases in this case, because of Pleurotus is not strong lactase producer.
Exactly. I have searched on this concept a little bit, not deeply so much, but I have searched that for lignin degradation, I used Trametis.
Yes, yes. By the way, Pleurotus can also degrade, but slower. Okay.
Yeah.
The easiest approach, okay, is you can compare between if you use Trametis first followed by Pleurotus or the whole process with Pleurotus. It could take longer a little bit, but Pleurotus is more acceptable as grass when animal feed.
Okay. So do you think that if I used pre-treatment chemical methods rather than using Trametis?
No, I don't recommend. Once you go to biological, just continue biological. Because maybe during chemical treatment, you're going to have some inhibitors produced to the Pleurotus itself.
So it will not grow Pleurotus enough at this concept?
I don't, it's not recommended usually to use a pre-treatment chemically and then we go biologically. Once you go biologically, just straightforward biologically, the only things when this cultivation, please keep your eyes on the moisture content. Because mushrooms in general or fungus, they need humidity above 60%. Because this process, I think you do solid state fermentation.
Okay, I got it. Thank you so much, Professor Hisham.
You're welcome.
There is any other questions? I think, yeah, I think there is no other questions. So,
before we end, I would like to again express my sincere thanks for the committee head by Prof. Abdulatif Hisham, because I feel today it's not a local program, it's international program. We have a people from Tunisia, we have a people that's making high visibility of the committee. And I think this is a very nice program and very successful. Keep continuing this, Prof. Hisham, Abdulatif Hisham. And I am very happy for being part of this lecture series, actually. And inshallah, I'm looking next time to be in person when I come to Egypt to meet you all, inshallah. And I would like just to say in Arabic, you will understand. مجمع الحبايب يا دكتور هشام الله يكرمك في كتير من ناس من سنين ما قابلتهمش.
ربنا يكرمك.
الله يكرمك.
إن شاء الله we have international conference in next October. And inshallah we will invite you to come to meet you in Egypt بإذن الله.
Inshallah.
Thank you all for coming to the Elite Scientist platform. Thank you, Dr. Hisham, for this amazing lecture. Thank you, Dr. Abdulatif. Thank you, all the committee members and all the attendees. Actually, we're really honored to have you all with us. And also would like to announce the coming lecture. Dr. Abdulatif would like to announce the coming lecture if you have the date already. Dr. Hisham.
I think the coming lecture, inshallah, it will be the next month with Professor Ahmed Zohari بإذن الله.
تمام. Okay, so next
and
month
you can say your lecture title.
Okay. So our next lecture will be after 1 month. We will announce it. Also in the same groups which you already seen, and you have seen this lecture, the announcement of that lecture. So we will announce it maybe in coming few days, maybe after the Eid al-Adha, inshallah. كل سنة وانتم طيبين. And this lecture will be how we can make alternative animal models by AI for biologists. So this will be the main topic, and of course it will be a lot of discussion and a lot of things about it. So we will be very happy to invite you all to attend this lecture, and we will announce it soon. شكرا لكم يا جماعة جميعا على حضوركم. بإذن الله تابعونا على نفس المنصات اللي انتوا شفتوا فيها الإعلان دوت. بإذن الله تعالى هنتم الإعلان على المحاضرات الجاية واللي بعد الجاية بإذن الله. وشكرا على حضور
إن شاء
الجميع.
الله إن شاء الله there is also other lecture about the genome editing in agriculture. This will be with by Professor Nagwa from Cairo University. Inshallah, after maybe in July it will be this lecture. شكرا لكم جميعا. ويعني أنا سعيد جدا جدا بالدكتور هشام. الدكتور هشام ده يعني أخ عزيز، وإحنا على فكرة إحنا عاملين شغل كتير مع بعض، وإن شاء الله بإذن الله نستمر، وبإذن الله يعني كل شهر ونفوت شهر كده ويبقى فيه محاضرة جديدة من البروفيسور هشام بإذن الله. شكرا جدا لحضرتك، وشكرا جدا للجنة، لكل أعضاء اللجنة. فشكرا لكم جميعا، وشكرا لأكاديمية البحث العلمي. شكرا.
شكرا جزيلا.
شكرا جزيلا دكتور هشام، أسعدتنا بالمحاضرة.
الله يخلي سعادتك يا دكتور.
شكرا هشام، دايما منور كده باذن الله في كل الدنيا. دكتور هشام ربنا يفيك حبيبي.
ربنا يخليك يا دكتور.
شكرا جزيلا دكتور هشام، محاضرتك كانت شيقة جدا وممتعة. ربنا يبارك في حضرتك، نورتنا. ربنا يبارك.
في رعاية الله يا فندم.
في امان الله. كل سنة وانتم طيبين جميعا.
وانت طيب سيادتك.
صحة وسلامة جميعا.
الله يسلمك.